Hello guys, this is Dr. Daniel and welcome to Sqadia. For more details about my profile, kindly visit sqadia.com. So today we are going to discuss about the insulin, glucagon, their metabolic effects, and we're going to discuss diabetes mellitus. So today's lecture outline would be the hormonal regulation and an introduction about the islets of Langerhans, insulin, their receptors, their metabolic effects, and the glucagon. And we're going to also study and discuss the summary of blood glucose regulation with diabetes mellitus. So to begin with, to clear up all the concepts about the hormonal functions, we should know, and their regulation, we should know a difference between anabolism and catabolism. What are they? So to maintain a balance, there is always a balance between the anabolism and catabolism. So catabolism is the formation of more smaller molecules from complex molecules for cellular reactions, for cellular respiration. Like for example, and if you can see in this diagram, the glycogen is converted into glucose. So a larger molecule is broken down into the smaller molecule. So likewise in anabolism, there's a synthesis of complex molecules from the combination of smaller molecules. So the smaller molecule is glucose, and it is building up or combining with few other molecules to form glycogen. So this is anabolic effect, and this is the catabolic effect. So in the same way, triglycerides, which are the smaller molecules, they are the more complex molecules. They are broken down to form more smaller molecules that are the fatty acids, and these fatty acids then build up together and they form triglycerides, creating an anabolic effect. So insulin, sex steroids, growth hormone, thyroxine are the examples of anabolism, and catabolism, glucagon, epinephrine, glucocorticoids, growth hormones, and thyroxine has the effect of catabolism. Now we're going to discuss a little about the pancreas. So pancreas are composed of two types of tissue. One is two types of tissue with one tissue having acini which secrete digestive via pancreatic duct into the duodenum, performing exocrine function. And there are islets of Langerhans which empty their secretion into the blood, performing endocrine function. So there are two types of glands. One are exocrine glands and the other are endocrine glands. So endocrine glands are those glands which release their secretions directly into the bloodstream, while the exocrine glands, they secrete their secretion through duct. So like for example, sweat glands. So islets of Langerhans, there are almost 4 types of cells that are present in the pancreas or in the islets of Langerhans. So number one is beta cells. These are 60% of the cells secreting insulin and amylin. Then there are alpha cells, which are 25% of the cells present in the cells of Langerhans, and they secrete glucagon. Then we have delta cells, which secrete somatostatin, and PP cells, which are the pancreatic polypeptide cells. So secretion of pancreatic hormones into the portal vein provides higher concentration of hormones to the liver. Like for example, when the glucose level is increased in the blood, so insulin is secreted and it directs the flow of the glucose into the liver, which is stored as glycogen in the liver, which we will be discussing in detail in further slides. So this is an anatomical picture of pancreas. You can see they are the acinar cells which secrete the digestive enzymes. Then we have the ductal cells. So we have the exocrine portion, and then we have the endocrine portion of the pancreas, which containing the cells, the four types of cells which we have discussed. Now this is the more cross-sectional view of the islet of Langerhans. We have all the four cells. We have beta cell which secrete insulin, then we have alpha cells which secrete glucagon, and then we have delta cell which secrete somatostatin, which is a peptide hormone which inhibits the secretion of insulin and glucagon. Then we have F cells, which are also called as PP cells, which secrete pancreatic polypeptide. So this is the islet of Langerhans cells, and each cell perform according to the metabolic function of the body. So insulin is a peptide hormone which is produced by the beta cells of the pancreatic islets. It regulates the metabolism of carbohydrates, fats, and proteins by promoting the absorption of especially the glucose from the blood into fat, liver, and skeletal muscle cells. So it is a polypeptide containing two amino acids, one 21 and the other one is 30 amino acid respectively. They are synthesized as pre-pro hormones. They are packed as granules in the Golgi apparatus, and they are released as free hormones with C-peptide or connecting peptide. Insulin is circulated as unbounded carrier protein. Half-life is 5 to 10 minutes, and they are 50% of insulin is metabolized in liver and the rest is metabolized in kidney. So the insulin has its metabolic effects. Number one is energy abundance. Influx of nutrients releases insulin, the carbohydrates, the proteins that from the diet we take in daily. So insulin is released with the source of from the source of carbohydrate and protein-rich stimulus. So these nutrients for energy source, and they perform the anabolic processes in the cell. We know what anabolism is. Anabolism is the synthesis of more complex molecules from the smaller molecules. So there is a storage of energy sources for the later use. There is a rapid uptake of glucose in the liver and amino acids and potassiums in the cells. There's intermediate action on protein synthesis, inhibition of protein degradation. There's an activation or inactivating enzymes. There's an increasing transcription with delayed actions on carbohydrates. The proteins and fat metabolism that occurs in seconds, minutes, and hours. So we know it's an energy abundance. Energy is required because because it is an anabolic. It has an anabolic effect. So there are more synthesis of the complex molecules. So more energy is required for the building of the smaller molecules to create a larger molecule. So energy is utilized. ATP is utilized. So autophosphorylation is the insulin has receptors. Insulin has two types of receptors, which we are going to discuss later in the later part of the discussion about the insulin receptor. So insulin receptor is a tetramer with two alpha subunits which are lying outside the cell membrane, and there are two beta subunits which protrude through the cell membrane into the cytoplasm. Once the insulin binds with the alpha subunit, there is a autophosphorylation of the beta subunit, forming a kinase pathway is activated. Phosphokinase pathway is activated, and they performing the metabolic effects, and they are creating the metabolic effects, performing the function. The regulation is registered with the autophosphorylation, and the effects of insulin is registered with it. So there are multiple phosphorylation of enzymes and proteins to occur after this process of autophosphorylation. So a signal transduction is achieved by autophosphorylation of intracellular domain of its own receptor. You can see once in this diagram the insulin attaches with, these are the alpha receptors and these are the beta receptors, and this is the phosphate bonds from where the kinase activity or the signaling pathway does occur. So once insulin attaches with the alpha receptor, there's an autophosphorylation of these beta receptors, and they creating a signaling pathways or kinase pathways with the phosphate 3 kinase pathways, and there is a cell growth proliferation performing their normal cellular function, which is the cellular growth, proliferation, gene expressions, synthesis of lipids, proteins, and glycogen, cell survival, and proliferation. So once the glucose level rises, so insulin drags the glucose into the liver, and they are stored there as glycogen. Glucose is converted into phosphate 3 kinase, which we're going to discuss later because of the autophosphorylation of this beta receptor. So after this formation of phosphate 3 kinase, what happens is there are carrier proteins, which are the glucose carrier proteins, which we call as GLUT4, GLUT4. As you can see, these proteins or these vesicles, GLUT4 receptors or vesicles containing receptors, they are transported to the cell membrane, and from where they get attached to the cell membrane, the receptors accept the entry of glucose, and so the glucose enters inside the cell and perform its function by converting it into pyruvate or storing it as glycogen in the liver. So we're going to discuss first the carbohydrate metabolism. So in muscle, insulin providing glucose, now its concentration gradient from blood into the cell. So glucose transporters on the cell membranes, which we have discussed, the GLUT4, are recruited by the cytoplasmic vesicles. There is a glycolysis and oxidation and are stored as glycogen in postprandial period and in exercise. In the fasting period and in the exercises, it usually stored as glycogen. So glucose is utilized and transported with the help of insulin into the skeletal muscles with the help of the carrier proteins that are GLUT4 after the autophosphorylation of the beta receptors. These GLUT4 then are taken to the cell membrane, and they carries the glucose inside the cell, especially in the postprandial period and during the exercises. So the glucose is stored as glycogen in the skeletal muscles. So in liver, after the phosphorylation of glucose to glucose 6-phosphate by glucokinase, there's a glycogen synthesis by activating glycogen synthase. So glucose is converted or phosphorylated to glucose 6-phosphate by glucokinase, which glucose 6-phosphate then converted into glycogen by activating glycogen synthase, and it directs the flow of glucose by glycolysis through glycolytic enzymes, which are phosphofructokinase and pyruvate kinase. So how the hepatic outflow of or output of glucose is decreased by these following factors. They inhibit the glycogenolysis by inhibiting glycogen phosphorylase. There's the exit of glucose is inhibited by inhibiting glucose 6-phosphatase. So we know that glucose is converted into glucose 6-phosphate. So it is the exit of glucose is inhibited by the glucose 6-phosphatase, an enzyme that prevents the exit flow of glucose from the hepatic cells. From the liver cells, they inhibit glycogenolysis by inhibiting the glycogen phosphorylase. So the breakdown of the glycogen, once the glycogen is breakdown, it is converted into glucose. So it inhibits the breaking down of glycogen. It inhibits gluconeogenesis. Gluconeogenesis is the formation of glucose by the amino acids. So, by decreasing of uptake of amino acids in liver, like if the amino acid, the protein-rich diet which we are eating, so it also stimulates the effect of insulin. So, amino acids are dragged to the liver for the formation of glucose. So, it prevents or it inhibits the entry of amino acids into the liver. Hence, the gluconeogenesis is inhibited. There are few gluconeogenic enzymes, which are pyruvate carboxylase and fructose 1,6-diphosphatase. It enhances the synthesis of fatty acids by converting glucose to pyruvate with the glycolysis process and forming acetyl coenzyme A, and secondly converting acetyl CoA to manylyl CoA. So there is no formation of keto acids, as since the fatty acids are formed, the synthesis of fatty acid, it means uh, it is forming, insulin is forming fatty acids by the help of conversion of glucose into pyruvate, which enters into the mitochondrial matrix, and where the electron transport of the Krebs cycle occurs, and there is the formation of acetyl coenzyme A, and ATP is released. So in adipose tissue, once the glucose is transported from the cell membrane into the cell, the glucose is converted into alpha glycerol phosphate, providing alpha glycerol for the esterification of fatty acids as triglycerides. So we came to realization that it is a process. Insulin has uh, has lipogenesis effect, and plus it inhibits lipolysis. So there's a formation of fatty acids. There is no oxidation of fatty acid, but there is a formation of fatty acids, and there is no formation of ketonesis. So it is anti-ketogenic at the same time. So how it is metabolized in insulin, how metabolization of insulin occur in brain, it has little effect. It directly depends on the insulin concentration. Glucose is permeable in the brain cells, and if the concentration is low, hypoglycemic shock with fainting, seizures, and coma usually do occur. Now we're going to discuss about the fat metabolism, and that is the adipose tissue. So we know it enhances the storage of fatty acids, inhibits the mobilization of fatty acids. So insulin inhibits hormone sensitive lipase, decreasing lipolysis of triglycerides. So esterification of fatty acid stored as triglycerides by forming glycerol, lipoprotein, lipase, and protein in the capillary walls, breaking circulating triglycerides into fatty acids to fat cells. So it has effect on lipogenesis, plus it inhibits the lipolysis and decreases the conversion or breaking down of the triglycerides. There is an esterification of fatty acid because of the formation of the alpha glycerol, which is formed by the pyruvate, which is converted into acetyl coenzyme A, and which then converts itself to melanin CoA. So in liver, the fat metabolism and the effect of the insulin is by the synthesis and inhibition of oxidation of fatty acids. So there is no oxidation of fatty acids, and there is decreased mobilization of fatty acids, and it increases the storage, so that the energy should be there or to be required for the further functioning of the cells. It enhances the synthesis of fatty acids, which we had discussed, from glucose to pyruvate by the help of glycolysis, forming acetyl coenzyme A, and then again converting into melanin CoA. The melanin CoA inhibits carnitine acyl transferase, which which inhibits the mobilization of fatty acids from the mitochondria and inhibits the oxidation and conversion to the keto acids. So we know that insulin is anti-ketogenic, has a anti-ketogenic effect. So protein metabolism of the insulin is, it is an anabolic hormone, limiting the rise of plasma level of certain amino acids, transports of amino acids from blood cells after the protein intake, and protein synthesis by transcription and translation of messenger RNA, inhibits catabolism of proteins and inhibiting the release of proteins from the muscle cells. We know that during the exercise and postprandial period, the amino acids, they are, the glucose is stored, and the amino acid is stored within the cell for providing an energy to the muscle during that specific period. So glucose, once its level rises in the blood, insulin drags the glucose into the liver, converting it into glucose 6-phosphate, which then converted into glycogen. It is stored as glucose is stored in the liver as glycogen. So the outflow of glucose into the blood from the hepatic cell is inhibited by inhibiting the gluconeogenesis and inhibiting the glycogen genolysis, and these two effects basically decreases the outflow of glucose from the hepatic cells directly into the blood. So it has anabolic effect, plus it has it inhibits gluconeogenesis, it inhibits glycogenolysis, and then it inhibits the oxidation of fatty acids, it inhibits the mobilization of fatty acids, and it stores the fats into the adipose tissue. Alpha glycerol is formed, and which converts itself into acetyl coenzyme A and then melanin coenzyme A, inhibiting the formation of keto acids. So it has an anti-ketogenic effect on the muscles, in the adipose tissue. So these are the metabolic effects. So glucose is converted by the glucose 6-phosphate, converting into glycogen, which can be converted back into the glucose, and glucose moves out of the liver into the adipose tissue and gets stored. In the same way, the fatty acids that are formed in the adipose tissue with the help of the alpha glycerol, it prevents the oxidation, the further acidification occurs, and keto acids are formed in the liver. And now these were basically keto acids. It basically it inhibits the formation on oxidation of fatty acids. Or the insulin inhibits the oxidation of the fatty acids to form the keto acids. The red arrows shows the stimulation, while the black arrows, they show the inhibition. So once the plasma level decreases, or free fatty acid decreases, keto acids decreases, and the amino acids decreases. These are the effects of the insulin on these four metabolic hormones, or the metabolic molecules. Now, glucose, once it is formed in the liver, they are transported directly into the protein, and amino acids are also stored in the protein. It prevents the outflow of glucose, and the amino acids both are prevented from the muscle, and they are utilized during the postprandial and the exercise period. There are major metabolic effects of insulin and the consequences of insulin deficiency. Likewise, in adipose tissue, we know it inhibits the oxidation and inhibits the mobilization of fatty acids. It accommodates more and more of the fatty acids. So it has the effect of lipogenesis and inhibits the lipolysis. So insulin effects stimulates the glucose uptake into the muscle, and the adipose cells have lipogenesis and inhibits lipolysis. The consequence The consequences of insulin deficiency on the adipose tissue is the elevated FFA levels. With the insulin effects, they inhibit the ketogenesis. The consequences of the insulin deficiency is the ketoacidosis, production of ketone bodies. Since we know that it is an anti-ketogenic it has anti-ketogenic metabolic effect, so it is an anti-ketogenic hormone which inhibits the ketogenesis, inhibits the formation of the ketone bodies, and once if there is any deficiency in the insulin, then there is a formation of ketoacidosis and forming production of ketone bodies. It stimulates the glucose uptake into the muscle, stimulates amino acids uptake and protein synthesis, inhibits protein degradation, and regulates the gene transcription. So consequences of insulin deficiency is muscle wasting. If there is no insulin, or there is a deficiency of insulin, then there is a wasting of muscle, that the muscle cannot store protein, and so it means it will go in the opposite effect. Like insulin is performing anabolic effect, then it is converted to catabolic effect, so they will be broken down of the more into the smaller molecules. The complex molecules are broken down into smaller molecules, and there is a muscle wasting then start to occur. So this is a quick summary of the insulin and its metabolic effects. Once the insulin gets itself attached to the membrane receptor, there is a signal transduction mechanism, and tyrosine kinase cascade reactions occur with the autophosphorylation of the beta receptors, and there's a glucose that is transported into cell, muscle, and the adipose tissue. There's a glycogen synthesis in the liver, adipose, and in the muscle. It inhibits the gluconeogenesis, glycogenolysis in the liver, and it activates the lipogenesis, which is in the adipose tissue and in the liver, inhibits lipolysis, inhibits the oxidation of the fatty acids in the adipose tissue. Then there is an amino acid cell transport into the muscles, into the liver, and in the adipose tissue. There is a protein synthesis from the muscles, from the adipose tissue and liver, and the liver tissues, and it inhibits the protein degradation, muscle, and probably other tissue. Once we before getting yourself confused with all these metabolic effects, you should be very much clear about the effects of anabolic hormones and the catabolic hormones. Once you are clear with the concepts of the anabolism and the catabolism, then the effects of the insulin and its metabolic effects will be much clearer and much easier for you. So up till now we have discussed about the exocrine glands, endocrine glands. We have discussed about the islets of Langerhans. We have discussed about the cells which secrete insulin, glucagon. Beta cells secrete insulin, which are the most in number, almost occupying 60% of the pancreatic cells, and they secrete insulin. And then we have alpha cells which secrete glucagon. Then we have delta cells which secrete somatostatin, which is a peptide hormone which inhibits the release of insulin and glucagon. And then we have PP cells, which are the polypeptide cells. Then we discuss about the insulin, its receptor. It has two receptors. One is the alpha, 2 units of alpha subunits, 2 alpha subunits, and 2 beta subunits. Once insulin gets itself attached with the alpha receptor, then there is the autophosphorylation of the beta receptors and performing a tyrosine kinase cascade reactions. 4 major effects are in liver. It is stored as glycogen. It inhibits the glycogenolysis. It inhibits gluconeogenesis, and in adipose tissue, it inhibits the oxidation of the fat cells. In inhibition of the fatty acids, inhibition of the oxidation of fatty acid. It is stored as fatty acids, and it inhibits basically lipolysis. or in liver and in adipose tissue it activates or it performs lipogenesis. Then in the muscle the protein and glucose is also stored. So up till now we have discussed about insulin. Now we're going to discuss about glucagon, its synthesis, its metabolism. And before jumping directly into the glucagon, we also should be discussing the control of insulin secretion, and that is the blood glucose concentration rises once it rises above the fasting levels, which are more than the 100 milligram per deciliter. That is the normal range. Of the blood glucose level, the insulin is secreted. Once it goes above the 100 milligram per deciliter, that is, it is moving above the normal range, that is close to 120, which is also called as the pre-diabetic period. The insulin starts to release from the pancreatic islets, dragging the glucose from the blood into the liver, into the peripheral tissue. So there's a negative feedback mechanism occur. So amino acids has also the synergistic effects with the glucose and stimulating insulin. Gastrointestinal hormones, gastric inhibitory polypeptide hormones, glucagon like polypeptide are released after the meal is taken, stimulating the insulin. Then we have cortisol and the growth hormone and the Cushing syndrome and the acromegaly, which causing the hypertrophy and exhaustion of the beta cells, so leading to increase in the insulin secretion, which antagonizes the effect of insulin on glucose uptake in the peripheral tissue. Then in sympathetic nervous system, it inhibits the insulin secretion. Parasympathetic increases the insulin secretion. Beta adrenergic stimulation increases the insulin secretion, whereas alpha adrenergic inhibits it. Now glucagon is a peptide hormone with a single chain of 29 amino acids. They are alpha cells of pancreatic islets of Langerhans, from where they are secreted and constituting almost 25% of the cells present in the in the islets of Langerhans. They are also known as the hyperglycemic hormone, tend to increase the blood glucose level. The blood glucose level, once it falls down from the normal range, then the blood glucose level increases after the release of the glucagon from the alpha cells. Main action is on the liver. They are synthesized as pre-pro hormones just like insulin, and they are packed as granules in the Golgi apparatus just like insulin, and half-life and metabolysis in liver is similar to the insulin, that is 5 to 10 minutes of the half-life, and the metabolysis 50% of the metabolysis occurs in the liver, while the rest is metabolized in kidneys. So these are the physiological actions we are going to be discussing about glucagon. So they are the physiological actions. Glucagon has its effect on glucose. It increases the blood glucose level. On fats, it decreases lipogenesis, and on insulin, it stimulates the insulin secretion. So it is totally opposite, or totally antagonizes the effects of the insulin. It is a catabolic hormone. Insulin was an anabolic hormone. So there will be a catabolic effects on the liver, on the fats, on the adipose tissue. So now we're going to discuss the metabolism in the liver. There's a binding of glucagon on the hepatic receptors. They activate the adenyl cyclase. There's a generation of second messenger cyclic AMP. There's an activating protein kinase A activation or deactivating number of enzyme of phosphorylation. There's a glycogenolysis by activating glycogen phosphorylase, releasing glucose into the blood. So we know that glucose is low in quantity and has a low level in the blood. So what happens is that the glycogenolysis occurs. There's a breaking down of the glycogen, so we are going to get a catabolic effect, the breaking down of the larger molecule into smaller molecules. So glycogenolysis occurs, which is inhibited in the insulin. When the insulin is secreted, now it becomes activated with the help of glycogen phosphorylase, and the glycogenolysis occurs, and there is releasing of glucose into the blood and inhibits glycolysis, increasing the glucose 6 phosphate levels, leading to increased glucose from the liver. So stimulates gluconeogenesis. With the help of amino acids, glucose is formed. The glucagon is ketogenic, inhibiting the acetyl-CoA transferase, acetyl-CoA carboxylase, inhibiting malonyl-CoA, thereby directing fatty acids to mitochondria for oxidation and ketogenesis. So, formation of ketone bodies occur by directing the fatty acids to mitochondria for oxidation. In insulin, we had it inhibited the mobilization of the fatty acid, and it inhibited the oxidation of fatty acids. While in the mitochondria, the fatty acids are directly oxidized to ketone bodies, so it has ketogenic effects. So these are the metabolic effects we're going to see about the glucagon. In this diagram, there's a glycogenolysis that does occur. There's a gluconeogenesis, secretion of insulin, inhibition of anabolism, because it has a catabolic effect. It is going totally opposite of insulin. And there's a fatty acid, increase in fatty acids and ketones. It means it increases or it activates lipolysis and inhibits the lipogenesis. There's a release of secretin, cholecystokinin. These are the two hormones, and the sympathetic activity. While in the insulin secretion, sympathetic activity inhibited the insulin secretion, while in glucagon, it increases the release of the glucagon, and parasympathetic activity has the opposite effect while inhibiting the release of glucagon. Once the insulin levels are high, then the glucagon levels start to fall down. So these are the stimulating factors. There's a decrease in the glucose and increase in the amino acids. While there was in insulin, there was increase in the glucose level, and there is an increase on the amino acids. While in glucagon, there are increase in the amino acids and decrease in the glucose levels. Stimulate the release of glucagon. It means that glucagon has its effect on the liver, while the insulin had its effect on the skeletal muscle, on the liver, and in the adipose tissue. So since amino acids entry, or it initiates the release of the glucagon, because it helps in the formation of the glucose, which is through the process gluconeogenesis. Now the factors which stimulate and inhibit glucagon. Glucagon is a controller. Glucose is a controller of glucagon and insulin. Any hypoglycemic condition increases the glucagon secretion. Hyperglycemic conditions increases the release of the insulin, and also the increased protein-rich diet which we take initiates or gives the stimulus to release insulin, and it controls. During the fasting and exercise, it increases the glucagon secretion because the glucose levels have fallen down. So glucagon secretion increases. The glycogenolysis and the gluconeogenesis, which the proteins which were stored before during the effects of the insulin in the skeletal muscle, in the liver, they start to reverse their process by the release of glucagon, performing glycogenolysis and gluconeogenesis formation, causing the release of the proteins and also the release of the glucose into the blood. And there's beta adrenergic stimulation, which we have discussed earlier, that it increases the glucagon secretion while it has the opposite effect on the insulin secretion. And alpha adrenergic stimulation increases the insulin secretion and it inhibits the glucagon secretion. Somatostatin, which is a hypophysiotropic or polypeptide hormone, it is synthesized from the delta cells of the pancreas, gut wall, and the hypothalamus. It is a 14 amino acid peptide simulated in pancreas with increase in the blood glucose level, amino acid level, and the fatty acid and the gastrointestinal hormones. It inhibits the gastrointestinal mobility, absorption, and secretion. It is an inhibitor of both insulin and glucagon. This is a summary of the blood glucose regulation. You can see in the hyperglycemic and the in the hyperglycemic condition, which is above the 120 milligram per deciliter of the normal blood glucose level. So once the beta cells of the pancreas, they simulate once they when the homeostatic levels are imbalanced, the blood glucose level rises and the insulin is secreted from the beta cells of the pancreas. The body cells take up more of the glucose and the liver takes up the glucose and store it as glycogen, and blood glucose level comes back or declines to the set point, to the balancing state. Once this is the regulation diagram of a blood glucose level when it becomes high in the blood. So once the blood glucose level is low, then the glucagon alpha cells, they are stimulated and they release glucagon, and liver breaks down glycogen by the process glycogenolysis and releases glucose into the blood, and the blood glucose level rises to set point stimulus for the glucagon release diminishes. So up till now we have discussed about the insulin and glucagon and their metabolic effects. Now we're going to discuss the last topic, that is diabetes mellitus. Diabetes mellitus is commonly referred as diabetes. It's a group of metabolic diseases in which there are high blood sugar levels over a prolonged period. So there are two forms or two conditions that are present, or two types of diabetes mellitus. There are type 1, which is diabetes mellitus, also called as insulin dependent diabetes mellitus. There is an impaired insulin secretion. Then we have type 2 diabetes mellitus, also called as non-insulin dependent diabetes mellitus, which is resistant to the metabolic effects of the insulin in the target tissues. So which is insulin dependent diabetes mellitus, that is the type 1 diabetes mellitus. There is an autoimmune destruction of the beta cells or by the viral infection. It usually occurs in the childhood. It is also called as juvenile diabetes. Hyperglycemia and increased glucose production by liver, that is the gluconeogenesis. There's a depletion of proteins due to decrease in the protein synthesis, and there's an increased catabolism and leading to glucosuria. There's an osmotic diuresis. There's a hypovolemia, hypotension, hyperosmolality of blood. They're causing the polydipsia and dehydration. Then we have hyperphagia, which is the weight loss and the loss of energy. There are acidosis leading to diabetic coma, rapid and deep breathing. Then we have hypercholesterolemia, which and atherosclerotic vascular diseases. So there's an increased glucose production by the liver. There's increased glucose production by the liver with the help of gluconeogenesis. There's a depletion of proteins, and it performs the catabolic increase in the catabolism. It means that the larger molecules are broken down into the smaller molecules. So more energy is in the process is made. So these are the effects of the type 1 diabetes mellitus. To keep this thing short, we should know that it is a juvenile diabetes. It usually occurs in the childhood. It is an autoimmune destruction by the of the beta cells by and also by the viral infection. Now we have type 2 diabetes mellitus, which is non-insulin dependent. They are more common in adults, and obesity is the prime cause. Insulin resistance, resisting the metabolic effects of the insulin in the target tissue. Beta morphology is normal, while in type 1 there was a destruction of the beta cells, while in type 2 the beta cell morphology is normal, but the insulin, the effects of the insulin, they are impaired. There's an insulin resistance. Hyperglycemia is just it causes hyperglycemia. There's an accelerated lipolysis, and ketogenesis usually do not occur because the normally insulin in the fat adipose tissue, it inhibits the oxidation of fatty acid, it inhibits lipolysis, and activates the lipogenesis effect, and inhibits the ketogenic or the ketone body formation. So it will go opposite, because since insulin is resistant in the target tissues, the acceleration of these two processes usually do not occur, but it does occur. So caloric restriction and weight reduction usually improve insulin resistance in the target tissues. So people with overweight start doing exercise, walking, jogging, and it because these physical activities, because of the exercising activities, the caloric restriction and the weight reduction usually improve the insulin resistance in target tissues. Insulin is usually administered at the later stages of this disease. So we're going to quickly go through with the diabetic metabolism. There's an insulin deficiency and glucagon excess. Once there's a glucagon excess, then there's an increase in the protein catabolism. There will be increased lipolysis. There will be decreased cellular glucose uptake. So hyperglycemia, glucosuria, osmotic diuresis, and electrolyte depletion occurs. And there's an increase in the plasma amino acid nitrogen loss in urine. There's a dehydration, acidosis, coma, and death. There's an increased plasma FFA, ketogenesis, ketonuria, and ketonemia because of the formation of the ketone bodies. There's an increase in the lipolysis because there's an oxidation of the fatty acids, and these fatty acids, they are converted into the ketone bodies. So there's a total catabolic effect. The glucose, which was decreased cellular glucose uptake. There was a hyperglycemic state. There will be glycosuria because insulin is resistant. Once the glucose level is high, so insulin cannot drag glucose back into the liver and cannot store this glucose as glycogen. So glucose level remains high and becomes a hyperglycemic state. There will be increase in the protein catabolism. Amino acids, protein is broken down into the amino acids, and these amino acids then are taken into the nitrogens that are lost in the urine. They increase in the plasma amino acids, and it leads to dehydration, acidosis, and coma and death. Then there are other types of diabetes mellitus other than type 1 and type 2. Gestational diabetes that occurs during pregnancies, and it resembles type 2 diabetes mellitus. Then we have maturity onset diabetes of the young, which is an autosomal dominant disease. Then we have pre-diabetes. The blood glucose normal is high, but not high enough to declare it as type 2 diabetes mellitus, usually in the range of 110 to 115. It is a state of pre-diabetes. Now the diagnosis of diabetes mellitus, if according to WHO diabetes diagnostic criteria, if the two, after taking any carbohydrate rich diet, after 2 hours of glucose checkup, the normal range is less than 7.8 8, which is less than 140 milligram per deciliter. So after in diabetes mellitus, after the 2 hours of intake of glucose, if it is more or if it is if it is less than 200 milligram per deciliter, then we declare it as diabetes mellitus, while the fasting glucose would be higher than the 126, must be lower than the 126, so to be declared it as diabetes mellitus during the fasting stages. Now, the treatment of diabetes mellitus, we have medications, metformin for type 1 and type 2 diabetes mellitus, which lowers the blood glucose level. Then we have angiotensin converting enzyme inhibitors. Usually, sometimes we perform surgeries, that is pancreatic transplant, in addition to the later stages with kidney transplant. So we're going to summarize the whole lecture. We discussed about the islets of Langerhans, the pancreatic cells, which we have discussed, which were namely beta cells, alpha cells, delta cells, and the F cells. Beta cells secrete insulin, and delta cells secrete somatostatin, and alpha cells secrete glucagon. We discussed about insulin. Insulin is a anabolic hormone. It has anti-ketogenic effect. It inhibits the oxidation of fatty acids and stores the fatty acids in the adipose tissue. It stores the glucose and proteins in the muscle during the postprandial and exercise periods, and it also inhibits gluconeogenesis. It is stored in the liver as glycogen by converting itself to glucose 6-phosphate. Then we have we discussed about the receptors, that those were 2 alpha subunits and 1 were beta. 2 were 2 beta subunit receptors. Once the insulin gets itself attached with the alpha receptor, there is an autophosphorylation of the beta receptors, performing the tyrosine kinase cascade pathways, and the whole metabolic effects of the insulin occurs. Then we discuss about the glucagon, which is the catabolic hormone, and we have discussed that it activates glycogenolysis, gluconeogenesis. Most of its effect is in the liver. There's an inhibition of anabolism, and there's an increase in the fatty acids and oxidation of the fatty acids, and ketone bodies are forming. So it has a ketogenic effect. Then diabetic metabolism, we have discussed about a diabetes mellitus. It can be there are 2 types of diabetes mellitus, that is the type 1 diabetes mellitus, which is the childhood diabetes mellitus, and then we have the type 2 diabetes mellitus, in which is insulin is resistant to perform its metabolic effects on its target tissues, and it is usually occurs in the adults. So the diabetic metabolism, which we discussed, that the insulin deficiency and there is a glucagon excess, there will be decreased glucose uptake into the liver, and so the glucose level is high in the blood. Plus there will be increase in the amino acids in the blood. Plus there will be increased lipolysis. So all these things lead up to dehydration, acidosis, coma, and death. So this we discussed about the islets of the Langerhans. We discussed about the insulin, their metabolic effects, and also discussed about glucagon and their metabolic effects, plus the regulation of insulin and glucagon secretion in the blood. And we discussed about a disorder that was diabetes mellitus. I hope I made my lecture clearer and you guys have understood it well. So thank you for watching sqadia.com.